Hi, this is Dr. Launen from Keene State College, and I'm going to be talking to you about how chromatin state can influence eukaryotic gene expression. We're going to define epigenetics, or actually give and consider a couple of definitions for the term epigenetics, and we're going to consider how chromatin state affects gene regulation in eukaryotes, thinking about the histone code and the enzyme that controls the histone code, and how altering the histone code can affect eukaryotic gene expression. We're going to consider one additional functional group that can also affect gene expression, additional to the one involved in histone. I'm going to talk to you today about two functional groups. One will be on or off histone proteins, and the other will be on or off DNA itself. And then we're going to think about the role of one of those functional groups in something called genomic imprinting, and actually talk about what genomic imprinting is. So the prefix epi means to stand over or upon something. And so epigenetics means above or over genetics. And here are two definitions, and I actually prefer the second, but that's actually my bias. So I'll give you the first, more common one. Epigenetics is the study of potentially heritable changes in gene expression that are not due to changes in the genetic code itself. So something you inherit that's not due to the sequence of a gene, which is, a little bit baffling to think about, right? Which is why I like this second definition, which I took from the Genetic Science Learning Center at the University of Utah Genetics Learning site. And it is: as an organism grows and develops, carefully orchestrated chemical reactions activate and deactivate parts of the genome at strategic times and in specific locations. Epigenetics is the study of these chemical reactions and the factors that influence them. So we're going to think about regulating eukaryotic gene expression, and we're going to tie that together into this idea of epigenetics. Before we do that, we're going to go back to an earlier concept covered in class, and it's just the basic idea of what does it mean to regulate gene expression. So you know what the genetic message is. It means a gene or sequence of DNA in the genome that encodes either a functional RNA molecule like rRNA or tRNA, or a messenger RNA molecule. So the gene always encodes a unique RNA. If it's messenger RNA, it is then translated to form a peptide. And one peptide can be a protein, or perhaps several peptides together will become a final protein. And then this protein is what actually is responsible for some measurable trait in the organism. And so this is gene expression here. And it has to be controlled, because if all the genes in the organism were on full bore all of the time, then you would have basically cellular death. And in fact, we see that's what cancer is really all about, is uncontrolled gene expression and therefore cell proliferation. So you can control or mediate gene expression at any of these stages. And when we studied the lac operon in class earlier this semester, we studied regulation of transcription. And we focused on a prokaryotic system, but we learned about activator proteins and repressor proteins, which can bind at or near the promoter and turn up or down the activity of RNA polymerase, thereby influencing transcription and ultimately gene expression. So the thing is that now we're going to talk about eukaryotic systems specifically, and we're going to talk about how regulating the state of chromatin, like is it in euchromatin or is it in heterochromatin? Is it relaxed and open, or is it tightly bound up? How regulating that state is another way to regulate transcription. So we're still focusing on transcriptional control, but we're talking about doing it by controlling chromatin state. So let's introduce you to the idea of the phrase histone code. So histones, you should remember, are small basic proteins that in combinations of 8 form a core around which DNA wraps as chromatin is formed. Each of these little balls on a string is referred to as a nucleosome. And you should know all those phrases, and this should not be new information for you at this point in time. And so here we've got chromatin, and down here we've got chromatin. Same chromatin at two different moments in time and in two different physical states. Okay? open and closed. Transcribable, not transcribable. And it's basically as simple as that, except for what makes the difference. Well, this is the first time I think you've seen these little pokey, tails that are coming off not the DNA itself, but the histones. And so the histones can have functional groups, small combinations of organic molecules that are shown in characteristic patterns, one of which is an acetyl group. So acetyl might be CH3CO, for example, which is acetate itself. So if you add acetyl groups to the histone proteins in a nucleosome, then they serve to push each other apart, which opens the chromatin and thereby opens up this DNA, making it ready for transcription. If you remove these acetyl groups, as you see in the picture on the top, then the nucleosomes can come together very closely, and then the chromatin itself is closed and not accessible for the enzyme RNA polymerase, and therefore not transcribable. So what controls whether there are acetyl groups on or off the nucleosomes, the histones that make up the nucleosomes? The enzyme histone acetyl transferase. So that is an enzyme that can put acetyl groups on or off, opening or closing the chromatin, turning transcription up or down. And this is arrows going back and forth to show you that this is a reversible or could be a reversible process. So when we say histone code, we're not talking about the genetic code. We're talking about the pattern of functional groups that are attached down here or not attached up here to the histones that make up the nucleosomes that make up the base structure of chromatin. All right, so let's put a little bit more complexity in here. So now let's think about regulating transcription and what you know about it. You should know about activator proteins and repressor proteins. Activator proteins can bind to promoters and thereby turn up transcription by helping RNA polymerase anchor and start. Repressor proteins can bind to areas like operators or other areas in eukaryotic genomes, and they can repress or inhibit the activity of RNA polymerase, thereby turning transcription down. You already know about that, okay? And all of that happens in eukaryotic systems as well. Now you also know about the role of the histone code. So, and I explained that role in the previous slide as we were looking at it. So now let's put those two ideas together, okay? So here is an activator protein, and it is just like bouncing around. It's sitting here for a moment, but it's not able to get to this promoter, okay? It's like not really doing anything to help this promoter out. It should be sitting down near the promoter, helping to pull RNA polymerase down, but this is all bound up, not accessible for transcription, not accessible for RNA polymerase. Now here's that enzyme I mentioned a minute ago, histone acetylase or histone acetyltransferase. These names mean the same thing, and it is influencing the histone code. So now we've got these little red acetyl groups. They've been added to the nucleosomes themselves. This cartoon sort of implies they're stuck on the DNA, and they're not. They're on the green histone proteins themselves. When the acetyl groups are on, it opens the chromatin. Now that activator protein is going to be able to help RNA polymerase. Picture RNA polymerase as like a big giant blob that can now come in and bind to this promoter and then transcribe this gene. And RNA polymerase, as it goes, is able to like unwind the nucleosome structure and break the base pairs between the double-stranded DNA that are not shown here. So in this way, the histone acetylase is influencing the chromatin state, making it open so the activator protein can do its job, or leaving it closed so the activator protein cannot. So let's add another functional group to the mix. So this is a methyl group, CH3, okay? And this is a nitrogenous base, as could be found on any nucleotide, but this particular one is the base for cytosine. And cytosines in particular are the substrate for DNA methylation enzymes that can add a methyl group. So this methyl group's been placed on this cytosine in the double-stranded DNA molecule. These little M's are showing us methyl groups that are attached to cytosine. And that changes the chemical nature of the DNA. In general, the more methyl groups you have, the less transcribable the gene is. So increasing methylation reduces transcription, decreasing methylation increases transcription. It's an increased relationship. And so methyl groups on the DNA itself can significantly affect gene expression. So let's put that together with another concept. So we just talked about histone modifications, right? And we specifically talked about acetylation, but note that phosphorylation might be another possible modification that affects the histone code. And so here's a nucleosome structure. Here our histone proteins are light blue in this figure. This is chromatin. Each of these is a nucleosome, okay? And so if acetylation has occurred on the histones, it will open up the chromatin, and the state of DNA methylation will actually influence the activity of the histone acetyltransferase enzyme that does or does not acetylate the histone code. So DNA methylation affects whether or not histones are acetylated. It affects the histone code. So here we've got two scenarios. We've got a gene turned on, transcription is possible, and we've got a gene turned off. Okay? The little red dots are showing methylated DNA. Okay? And the little white dots are showing where the DNA is not methylated. Okay? So what's happening, and here are histones, okay? Like little tails coming off the histone proteins. And so what will happen is the gene will be switched on when the chromatin is open. That'll happen when it's unmethylated and when the histones are acetylated. So we've got acetyl groups on the histones, we've got methyl groups off the DNA. That turns the gene on. In contrast, when the acetyl groups are off the histones and the methyl groups are on the DNA, then we've got a closed or off situation and transcription cannot occur. So these mechanisms can all work together to control or influence transcription. Now, the last thing that we're going to consider as we think about epigenetics, or the chemistry that can affect DNA that is different from the sequence of DNA itself, right? What functional groups are on histones? What functional groups are on the DNA itself? We thought about acetyl groups on histones and methyl groups on DNA just now. Now let's think about a concept called genomic imprinting. So genomic imprinting is the idea that by adding methyl groups, or CH3s, onto a chromosome region or a gene on a chromosome during gamete formation, so as you build sperm or eggs, depending on your sex, you can tag a gene or switch it on or off. So if, as you make eggs, you tag a gene that's going to end up in your egg and you turn it off, then when you transmit the chromosome that carries that gene to your offspring, that gene will be permanently off in the offspring, which is pretty freaky because that means the only active copy of that gene will be the copy that came in from dad, which makes you actually haploid at a particular position of the genome when you think about it from a gene dosage point of view. So if the gene is being switched off in the mother as the egg forms, then it's called maternal imprinting. If the gene is switched off in the father as the sperm forms, then it is called paternal imprinting. But this is something that happened as your mom or your dad formed sperm or eggs, actually vice versa, right? Eggs or sperm. And then you inherited a copy that was of a gene that was permanently switched off. And this actually affects humans in a variety of ways and is the underlying biology that explains certain genetic conditions. So with that, I will finish discussing the topic of eukaryotic gene regulation through epigenetics.